up till now in these mini lectures on translation, it is very, very simplistic. One mRNA, one ribosome, one polypeptide. Done. That has been done, you know, just to make things easy for the artist who's drawing the figures. It's been done to make it easy for you students when you're looking at this to visualize and picture everything that is occurring. Turns out reality is a lot more complex. What we know from studying electron micrographs, transmitting electron micrographs of the interior cells during, active translation, that it is not one ribosome per one messenger RNA. In actuality, it's what is referred to as polyribosomes, numerous ribosomes per mRNA. How many? As many as can fit on. You see at the bottom there, that picture in a bacterial cell, one messenger RNA that is studded with ribosomes. What we know happens is as soon as the messenger RNA is being made, remember there's no cap, no introns, exons, we're talking bacterial. As soon as it starts being made and it's long enough, the first ribosome will hop on. And as it's moving down, as the messenger RNA gets made, a second one hops on. And as those two move down far enough, a third, and then a fourth, and then a fifth, and along comes a sixth, and a seventh, and an eighth, and ninth, and they're marching down. Hit the 3' end, first ribosome falls off, then the second one falls off, then the third, then the fourth. But as long as that messenger RNA is there and it remains stable, more and more ribosomes will just keep hopping on. So, in essence, one gene gives you one messenger RNA, which will result in 2, 6, a dozen, 2 dozen polypeptides being translated, being synthesized. Could be more. It all depends on what that gene was, and again, how stable that messenger RNA is and what's going on inside that cell. So it's all, for bacteria, time is of the essence. Okay? Their metabolisms are on super high. Especially, when they're on super high because there's lots of nutrients around. Got to remember, bacteria are dividing every 30 to 60 minutes. Staphylococcus aureus, E. coli. You have one bacteria, wait 30 minutes, binary fission, now gives you 2. Wait another 30 minutes, instead of 2, you now have 4. Their metabolisms are going so fast that they cannot wait. One messenger RNA, one ribosome, one polypeptide. No, everything is streamlined. So, here's the DNA. As the RNA polymerase, right here, is moving along, got that little transcription bubble going. As it's moving along, it's making the messenger RNA. As the messenger RNA gets long enough, a first ribosome will hop on. And that just keeps going and going. As the polymerase starts making longer and longer messenger RNA, now gets long enough, a second, then a third, then a fourth, then a 12th ribosome hops on. How do we know this? Well, it's up here. You look up here, this is an electron picture. Electron microscope picture, you can see the strand of DNA, RNA polymerase, And then all of this you see down here, all of this you see down here are ribosomes translating on a growing messenger RNA strand. We like to think, oh, us eukaryotes, because we got the nucleus and everything, and the chromosomes, the 5' cap, the tails, the introns, exons, all that, you know, we're more dynamic. Well, no, as you've seen the previous couple of slides, prokaryotes have a lot going on. Okay, they may not have the organelles and all that stuff, and all those other whiz bangs that we have, but their transcription translation. I mean, if you look at that, you know, that set of, on that last figure, one length of DNA, which probably had one or two genes, say just one gene. One gene had, you know, handful of RNA polymerases moving down it, and the growing messenger RNA that's being made by each of those polymerases is going to have, you know, a dozen or more ribosomes, right, you know, moving down it, translating to make the polypeptide. One signal calling for the making of one protein, the expression of one gene, sorry, and you ultimately could end up with dozens, if not hundreds, of copies of that one gene product polypeptide. So, prokaryotes, not as advanced as us eukaryotes, but there's still a lot of complexity, a lot of dynamic actions occurring. You look at ours, everything going on with ours. Signal calls for the initiation factors to bind to the promoter, which will then recruit RNA polymerase. RNA polymerase will transcribe, making a precursor mRNA. Precursor mRNA, that RNA transcript gets processed, 5' cap, poly A tail, introns spliced out. That mature messenger RNA gets trafficked out to the cytoplasm. Small subunit binds at the 5' cap. Initiator tRNA binds at the first AUG near the 5' end. Small subunit initiator tRNA will then recruit in large subunit. You get the elongation process of the growing polypeptide to get the end, the termination signal, and Bob's your uncle.